Hello everyone, this is Dr. Sudha, your mentor for biology. So today we are going to discuss a very important topic from class 12, that is lag operon. First of all, let us understand the meaning of lag operon. Lag operon has two words. One is lag and another is operon. Lag word is from lactose, and lactose means milk sugar, and operon means there is a single switch for controlling multiple bulbs. So here I can write that operon is a common promoter. It is a common promoter for controlling many genes. Common promoter is controlling many genes. From this word you can understand that for lactose digestion, means lactose has to be converted into glucose and galactose, and for this there must be a mechanism that is existing in prokaryotes. And as you know, prokaryotic genes are, these are polycistronic genes, means one gene is controlling many, one gene is controlled by, one segment of DNA is controlling many genes. It is controlling multiple genes, means it is the promoter where the RNA polymerase will bind, and from that binding of RNA polymerase, transcription will occur. Transcription will give rise to mRNA. mRNA will be helping in the translation, that is forming proteins. So here we have two conditions. And who gave this lag operon? This was given by two scientists, Jacob and Monod, and it is existing only in prokaryotes. So only prokaryotic cell, that is in bacterium, this mechanism is operating. Why it is operating? For the lactose digestion into glucose and galactose, so that a bacterium can cover its energy requirement with the help of glucose. So there occurs two conditions. Either in the absence of lactose, how this mechanism will perform, and in the presence of lactose, how it will perform. That means this operon is a type of inducible operon. Inducible operon means there is required some inducer to make it on or to make it off, and that inducer in this case is lactose. So I am taking here the two conditions, that is in the absence of lactose, how it will perform, and in the presence of lactose, how it will perform. So I am taking here two conditions. First condition is in the absence of lactose, how it will perform. So suppose it is a bacterial membrane and surrounding it there is no lactose, means there is no inducer. And here you have to remember this polycistronic genes, means one DNA segment having many genes, P, I, P, O, Z, Y, A. P, I, P, O, Z, Y, A, that is P, O, Z, Y, A. You have to remember this sequence. And before starting, let us understand what this P stands for. P stands for promoter. For promoter, the role of promoter is it provides the site for RNA polymerase to bind. What do you mean by I? I means, you can simply remember, it is inhibitor, or the most appropriate term is repressor. And these are the genes, basically these are the genes on one segment of DNA. What this I gene will be doing? It is forming the repressor protein, that is repressor gene. And this repressor gene formed by this repressor protein will be binding on the operator site, so that the transcription of the structural gene Z, Y and A, these are the structural genes. Structural genes means they are, after transcription they will be forming some enzymes, that is Z is forming beta galactosidase. What Y is forming? Y is forming permease, and A is forming transacetylase. Beta galactoside is the main enzyme for the breakdown of lactose into glucose and galactose. P, as the name is indicating, that is Y gene is forming permease, and as the name is indicating, permease means increasing the permeability of the bacterial membranes so that inducer can diffuse in. Here inducer is lactose. And A is trans-acetylase that activate both these beta-galactosidase and that permease. So we are taking the condition when it is absent. So after understanding this PIPOZYA, now there is no lactose in the medium, and it is obvious that cell will not destroy its energy in forming these enzymes to avoid the energy consumption. These ZYA are not transcribing to form any protein as there is no lactose, no inducer. So means in the absence of lactose, it is in the off position. Why it is in the off position? Because inducer is not present here. So this, if RNA polymerase will come and bind on this promoter site of repressor gene will transcribe to form mRNA. mRNA will form repressor protein, and this repressor protein will having some sites, active sites, by which it can bind on this operator site. This operator site is blocked by this repressor protein, and if this operator site is blocked, then this RNA polymerase of this operator site will not be able to move further, and these Z, Y and A, they won't be able to transcribe. That is, there is no transcription. That means there is no protein formation, means no enzyme, and there is, this system is in the off mode. That is, no energy is wasted by the cell if there is no inducer or lactose is present. That is, it is in the off position. Now take an example of when this is in the on position, or you can say in the presence of lactose. Second condition I am taking is in the presence of lactose. Make the same sequence, that is PI, PO, Z, Y and A. Right? Now what will happen if the lactose is present here? Lactose is present in the medium, and some lactose will get diffused inside the bacterial cell. How? Because of permease. Now students ask this question that if permease is synthesized by this Y gene, then how it has come here? So we can mention here that some permease is already present in the cell, that is allowing the lactose to enter in the medium. This lactose, when it enters in the medium, and meanwhile this mRNA of repressor gene, means repressor protein have formed. This is repressor gene has transcribed to form repressor protein, and this lactose will get bind, or you can say repressor protein now have more affinity for lactose. It will bind with this lactose, and it is not able to block the operator site. So this operator site is free now, right? Now when the RNA polymerase come, operator site is free. It is able to move freely. Z, Y and A, they will be able to transcribe, and after transcription all the three enzymes are formed. That is, Z will be forming beta galactosidase. What Y will be forming? Permease. And what trans-acetylase is synthesized by A, and that is gene A. All these three enzymes, they will be working for the digestion of that lactose into glucose and galactose. So with this will get on, this mechanism will get on, and the cell will digest its lactose to get glucose. If already glucose is present, then also this system will remain off, because cell do not require any energy for its survival. So with this is the lac operon explained in a very simplified way. Thank you.